Sure. Good morning, everyone. Today we're going to learn about electrophysiology. Electrophysiology is a branch of physiology that studies the electrical system of biological cells, tissues, and the techniques that facilitate the flow of ions. Generally, electrophysiology is divided into two: intracellular, which involves measuring current in the interior of a cell, and extracellular, which involves measuring current in the exterior of a cell. There is also neuronal electrophysiology. This is the study of electrical systems of biological cells and tissues in the nervous system. We move on to how electrophysiology is related to neuroscience. As we all know, neuroscience is the scientific study of the function, anatomy, and molecular biology of the nervous system. Therefore, voltage change in the brain helps us understand how complex information is processed through signals from the subcortical limbic structures to the hippocampal formation to form memory and cognition. Its relation is also seen in several additional tools which supplement research, including gene transfection techniques for live cell imaging of cell compartments, microfluidic devices to amplify neuronal signals and control neuronal growth, and more traditional tools such as immunohistochemistry and intracellular patch electrophysiology. Lastly, by comparing the current flow of cells to tissues of a case study to that of a healthy person, we can detect functional disorders of the nervous system. The techniques used are different for sensory nerve cells and motor nerve cells. When the sensory nerve or motor nerve cells in the brain or muscles process signals, they make electrophysiological examinations possible.
So these key terms are essential to understanding how electrophysiology research takes place. Neuronal cells have intracellular fluid and extracellular fluid, and it is the difference in ion charge of these fluids that creates an electrical potential. This electrical potential is what electrophysiology research is measuring. When ions are moving from intracellular fluid to extracellular fluid or vice versa, it creates a current that is measurable. The movement of ions is influenced by the amount of channels that are either opened or closed, creating a membrane resistance. These two factors, current and membrane resistance, allow for the measurement of voltage using Ohm's law, which is current times membrane resistance equals voltage. This measurement is the basis of electrophysiology. When the ions flow from intracellular fluid to extracellular fluid or vice versa, it changes the resting potential of -70 millivolts, either positively or negatively. If it changes positively, then the resting potential depolarizes. For example, instead of the intracellular potential being -70 millivolts, it would now be -60 millivolts. If the potential changes negatively, it is experiencing hyperpolarization. So the potential changes from 70 millivolts to -80 millivolts, for example. The magnitude of this change to the resting potential is equal to the strength of the stimulus. Synonymous with depolarization and hyperpolarization is excitatory postsynaptic potential, EPSP, and inhibitory postsynaptic potential, IPSP. EPSP is when the membrane potential becomes more positive, and IPSP is when the membrane becomes more negative. EPSP and IPSP generates an overall understanding of electrical signaling within the neuron. An action potential occurs when the neuron depolarizes to -55 millivolts, in which this voltage change is rapid and causes the voltage change to signal throughout the axon.
In order to understand electrophysiology research and how it came to be, it is important to note its origins as well. There are three important figures within the domain, and their contributed work have created an impact. Italian physician who was named Luigi Galvani is the author of De Verbis Electricitatis and Motus Colorali. It is a memoir that consists of the theory of animal electricity, which is also known as galvanism. He began his experiments on animal electricity in 1791. Later on, Italian physicist Alessandro Volta was inspired by his memoir and desired to perform further research with Galvani. These two figures have been the first to record bioelectrical signals, which has opened doors for future studies within electrophysiology. Further on, Guy Fontaine was a French cardiologist and electrophysiologist who was considered a pioneer in electrophysiology. Fontaine was one of the first to contribute to pacemaker technology and perfected his skills by receiving patients with arrhythmia problems. Throughout his career, he was able to publish the essentials of cardiac pacing alongside with his peers and mentors. The theory of animal electricity was done by dissecting a frog and utilizing its nerves to record electrical signals. This was done in order to achieve a muscle activation on an animal that is no longer living. For the experiments, Galvani used copper and zinc and connected its muscles and nerves through a metal conductor. As summarized by Bresadola, one of the writers of Journal of the History of the Neurosciences, a particular form of electricity exists between the interior and exterior of the muscle fiber, while the nerve fiber would penetrate inside the muscle fiber, allowing for an electrical flow. This leads to muscle contraction, and Galvani's memoir of the experiment has been read by many scientists, and Volta was particularly interested as he taught physics at the University of Pavia. Volta conducted further experiments with Galvani and believed that electricity was consistently circulating through the organism and produced contractions only when there was a disturbance or disconcernment in the harmonious circulation of the electric fluid within the organs of the animal. Volta has also written his own memoir called Prima Volta and illustrated the electrical imbalances present within the animal are due to an intrinsic electricity. Throughout the experiments, Volta and Galvani often did not see eye to eye for the following reasons. Galvani's main purpose was to observe the biological mechanism coming in contact with electricity, while Volta was more concerned about the physics of electricity within an organism. These experiments revealed the metal conductor, which is the external voltage source, did not cause the leg to contract. Rather, it was the nerve and muscle tissue, which was an internal generator of electrical energy. In all, Galvani's experiments demonstrated that the nervous system delivered animal electricity to muscle tissue. Further on, Guy Fontaine was a French cardiologist and electrophysiologist who had a background of electrical engineering alongside his medical degree. In the 1960s, he began his study within the field of electrophysiology, and he received his doctoral degree for his work entitled Contributions of Electrical Stimulation to the Human Heart. Thereafter, he was one of the many contributors in the advancement of pacemaker technology, and by using intercardiac leads, he was the first to implant permanent pacemakers. He performed and perfected epicardial mapping along with Dr. Robert Frank, which has led them to observe epicardial delayed potentials within their patients. Dr. Fontaine, Frank, and Girardon used handheld probes to map reentrant circuits in patients with ventricular tachycardia during surgery in order to disrupt the reentrant arrhythmia by ventriculotomy.
I will be going over current electrophysiology preparations and how they are scientifically organized. There are two main categories of preparing neuronal tissues, either in their natural environment or in a controlled experimental setting. The origin of in vivo and in vitro is Latin, in vivo meaning within a living organism, and in vitro meaning within the glass. In vivo preparations are consistent with the entire organism and its neuronal electrical impulses. And how it normally works is electrode, tetrode, or multi-electrode is inserted into the brain to make extracellular recordings. Intracellular recordings are less common because it is extremely difficult to place electrodes or patch clamps correctly. And if you guys don't know what patch clamps are, in the next portion of slides, Brianne will expand on the concept of patch clamps. And then transitioning to in vitro preparations, they provide very precise measurements of neuronal physiology due to their laboratory environment. And one of the more important factors for in vitro is the content of the bath solution and their ions. This is imperative because as ion concentration gradients change, so does the neuronal electrical activity. Both in vivo and in vitro can be divided into further subcategories that we'll explore in the following slides. The in vivo categories oppose each other, as you can see. So firstly, an acute recording is a process that runs for mere hours, whereas a chronic recording can range anywhere from multiple days to multiple years. The main benefits of acute recordings are the limited time and resources required. Researchers are more likely to be funded for acute recordings based on those reasons. And then going to chronic recordings, their biggest strength is their ability to collect a variety of data by surgically implanting electrodes into an animal's brain. And when animals are under anesthesia, researchers are able to expand on electrical impulses going to and from specific neurons. It is important to be cautious of the effects that different anesthesias can have on an animal's neuronal properties, such as excitability and neurotransmission. Animals being recorded while fully conscious are trained to perform certain behaviors. Once the animal has learned a behavior, electrodes are inserted into the brain to be able to discover specific neurons that are active depending on the task. And yeah, so partly due to heterologous expression systems, electrophysiologists can now better understand the reasoning behind neuronal changes with electrical properties by intentionally introducing a foreign gene to encode a specific ion channel. Today, Xenopus laevis oocytes, Chinese hamster ovary cells or CHO cells, and human embryonic kidney or HEK293T. These cells are commonly examined systems for heterologous expression systems. And then primary cultures, they are directly extracted from organisms. They come close to the accuracy of in vivo recordings. These are especially helpful when comparing for neuronal physiology of different populations. A good example of this would be examining regions wild horses versus domesticated horses. Slice cultures then are used within a day of extraction, and samples measure out to be roughly 200 to 500 micrometers thick. And due to its small size, obtaining data from the inner brain becomes less complex. It is easier to conduct intracellular recordings due to the lack of movement, since there is no heart pumping blood into the slice, as well as the fact that the sample is so thin. And then bath fluids are designed to keep neurons alive with the proper balance of certain ions and nutrients. And pharmacology favors this method because drugs can be applied to the sample and the effect in both pre and postsynaptic circuits are observable.
So for electrophysiology techniques, there are, they differ by the recordings of electrical current changes depending on where the electrode is placed. So extracellular recording is when the electrodes are placed outside the neuron of interest in the surrounding fluid. This technique is easy to use and does not require precise positioning of the electrode. It can also measure several extracellular action potentials at once within the brain. The main disadvantage to this technique is that it can only measure action potential and not EPSP or IPSP. Intracellular recordings record small graded potentials and localized change within the neuron. So the electrode is placed within the neuron of interest. And instead of recording action potentials, it records EPSP and IPSP. This has become an outdated electrophysiology technique and has been replaced with the patch clamp technique. The patch clamp technique has 4 different modes: cell attached mode, whole cell mode, inside out mode, and outside out mode. We will get into more detail about how these techniques functions in the next few slides.
Extracellular recording techniques are relatively simple and easy to conduct, and they don't require the same level of precision as the intracellular and patch clamp recording techniques. When an action potential occurs, changes in membrane voltage cause localized short-term differences in potential on the outer surface of the active neuron. Thus, by measuring the potential difference between the tip of a recording electrode and a ground electrode placed extracellularly, action potentials can be detected in the extracellular space near the membrane of an active neuron. Extracellular action potentials are characterized by a brief alternating voltage difference between the recording and ground electrodes. A disadvantage of extracellular recordings is that it is impossible to measure localized potentials, meaning they cannot report detailed information about subthreshold potential changes. Intracellular recordings identify slight graded changes in local membrane potential caused by synaptic events. To measure the potential difference between the end of the intracellular electrode and the reference electrode positioned externally to the cell, intracellular recording involves impaling a neuron with a micro electrode. It's important to note that an action potential measurement is different for extracellular and intracellular recordings. For an intracellular electrode, the difference in potential between an electrode and ground increases and then returns to the baseline, whereas for an extracellular electrode, the potential difference first decreases, then increases before returning to the baseline. Intracellular and extracellular techniques also use different units of measurement, with intracellular electrodes measuring potential differences in millivolts and extracellular electrodes measuring considerably smaller potential differences in microvolts. Patch clamp techniques utilize a micropipette, which is basically a small glass capillary with an internal diameter smaller than that of a cell. The micropipette is used to make contact with a small area or patch of neuronal membrane. When suction is applied to the back of the pipette, the seal becomes so tight that ions can no longer flow between the pipette and the membrane. This means that when a single ion channel opens in the patched membrane, those ions are forced to flow into the pipette. The resistance of the seal must be extremely high in order to make sure that none of the current leaks out to other regions of the membrane. This creates a small electrical current that can be measured with a sensitive amplifier. This arrangement is typically known as the cell-attached mode, and it allows the researcher to study single ion channels. Small technical modifications to this method result in 3 other recording variations of the patch clamp technique: whole cell inside out and outside out recordings. Whole cell recording allows measurement of electrical potentials and currents from the whole cell, hence the name. This occurs when the membrane patch inside the pipette is briefly disrupted due to the application of strong suction, causing the interior of the pipette to become continuous with the cytoplasm of the cell. This also allows diffusional exchange between the pipette and the cytoplasm, thus providing a convenient way to inject substances into a patched cell. Inside-out recording and outside-out recording both stemmed from the finding that once a tight seal has been formed between the micropipette and the membrane, little pieces of membrane can be pulled away from the cell without disturbing the seal. Inside-out recording involves retracting the pipette while it is in the cell-attached configuration, which causes a small vesicle of membrane to remain attached to the pipette. Once the tip of the pipette is exposed to the air, the intracellular surface of a small patch of membrane is exposed. This configuration allows researchers to measure single channel currents and also the ability to change the medium exposed to the intracellular surface of the membrane. Outside out recording involves retracting the pipette while it is in the whole cell configuration, which creates a membrane patch that reseals to expose its extracellular surface to the bath solution. Because the bath solution can be easily manipulated during recordings, this configuration also allows researchers to study how extracellular chemical signals, such as neurotransmitters, influence channel activity.
One of the limitations that can be experienced within electrophysiology in regards to transcranial magnetic stimulation and transcranial current stimulation would be that sometimes a site in the brain can be effectively stimulated, but the readout of that site, the indicative statistical measurement, can either be not sensitive in general for the induced neuronal effects, or the readout in general is lacking statistical power to detect these stimulations. One approach to fix this issue with the readout was to introduce the TMS-MEEG or TMS-EEG studies. TMS is used to target nodes of the dorsal frontoparietal attention network, while the EEG and MEG were utilized to assess the altered top-down impact on oscillatory neural activity in the occipital cortex. Another limitation that can hinder the readout of non-invasive transcranial stimulation would be that the target site is simply not getting stimulated. This can happen because sometimes the stimulation gets picked up in different sites of the brain or accidental stimulation through the wrong part of the brain. Even sometimes when doctors target an area of the brain for stimulation, it often can affect more distant areas of the brain. But standard montages with 2 electrodes generate a rather cluttered stimulation pattern with several field peaks between electrodes. This can mean that transcranial current stimulation can sometimes be touch and go with what works and what doesn't. Researchers and doctors who discovered this phenomena support the future innovation of individualized electric field calculations and more precise high-definition transcranial stimulation. They believe that by doing this, it will clear up the confusion and misfiring of activation of brain areas that are not being tested or weaken stimulation of a brain area.
The benefits of neuroimaging in electrophysiology show that they can inform non-invasive transcranial brain stimulation and provide information as to where, when, and how we can optimize stimulation to the brain. It can also provide a readout for the neural changes included by non-invasive transcranial brain stimulation. Michelle Dahan of the Institute of Child Health notes that in the future of electrophysiology, it might be practical to target multiple sites in a single experiment session by switching different transcranial current stimulation electrode montages while still retaining the spatial focality that has been demonstrated for multi-channel transcranial current stimulation, meaning that by moving around the patches on the skull while still maintaining the same focal point of the brain, we may be able to yield better and more accurate results from seeing the stimulation of the brain part coming from different directions. It has been shown that multidisciplinary research in the areas of biology, medicine, biochemistry, electronics, and physics has been very successful in the development of electrophysiology. Combining multiple disciplines of science together is beneficial for current and future research because it utilizes techniques from multiple domains to optimize the procedures and discoveries of electrophysiology, while broadening the horizons of disciplines being incorporated into electrophysiology. Through finding what works and what doesn't, it ultimately makes electrophysiology as a whole more precise and effective. Through these systems, doctors and researchers can then boost the therapeutic potential of non-invasive transcranial brain stimulation.
In conclusion, electrophysiology is an important tool in the diagnostics of various conditions in otherwise very delicate organs and tissues. Combined with the right disciplines, the process of electrophysiology will only become more precise and efficient. From the discussion above, electrophysiology is not only an important but also a necessary tool in the future of modern medicine. Thank you for watching and have a good day.